All right, so this is the second lecture for chapter 32. In the previous lecture, we went over some of the principles of genetics, and in this lecture, we're going to go over the different modes of inheritance and the Hardy-Weinberg equilibrium. All right, so this is a list of the different modes of inheritance, and we're going to go through each of these individually on the subsequent slides. So for each of these modes of inheritance, we have a sample pedigree, as you can see here. And these sample pedigrees are really important because one, they can show up as answer choices on exams. And then the other thing is, beyond that, beyond the exam, is they're really helpful for understanding the different features of each of these modes of inheritance. It's an illustrative example, if you will. And some things to pay attention to is, for each of these modes of inheritance, is do they affect multiple generations? Do they skip generations? Is one gender affected more so than the other, or are both genders affected equally? And so those are just some things to pay attention to. So we're going to start out here with autosomal dominant. This does affect multiple generations, so therefore it usually does not skip generations. Both genders are equally affected, and disorders that exhibit autosomal dominant are actually pretty rare in their population. So if we come over here to the sample pedigree, just a little key here. So the squares are males, and then the circles are females. And then if it's blank or white, that means that the individual has a normal phenotype or is healthy, does not have the disease. Or if it's filled in with black, that means the individual has inherited the disease. Some examples of diseases that exhibit autosomal dominant inheritance are neurofibromatosis 1, Huntington's disease, and then Marfan syndrome. So if we come down here and look at the pedigree, we look at the first couple here. So the mother is affected, so she would be a heterozygous. And then the father is healthy, so he would be homozygous recessive. And so for a autosomal dominant disorder, you only need one dominant allele to end up exhibiting the disease. You don't have to have both copies. Certainly if you have both dominant alleles, so if you're both capital A, the individual would be affected as well, but not required. And so what happens is if you have one parent that's affected and has children with another parent that is not affected, we can draw a Punnett square here to show the breakdown. And Punnett squares are important because they really illustrate where these percentages come from versus having to memorize them. You can actually see the breakdown yourself. And so if you look at the Punnett square here, 50% of the children are going to be heterozygous, meaning they're going to be affected by the disease, so as we say here. And then 50% are going to be homozygous recessive, meaning they will not be affected by the disease. And again, these Punnett squares are really important because worst case scenario, if you forget the percentage on the test, you can always just draw this out in the margin or on a scratch piece of paper, and then you can get the answer that way. So if you look at this couple's children, so they had 4 children, and half of them were affected, and then half of them were not. So half were homozygous recessive and half were heterozygous. Now, if you have a couple like this where both parents are affected, so they're heterozygotes, and we draw our Punnett square like this, if you look, 3/4 of the children, or 75%, are going to be affected because they at least have one dominant allele, and in this case they have 2 dominant alleles. So all 3 of these scenarios will be affected, and then they have a 25% chance of being normal, meaning they'd be homozygous recessive. And you look at this is illustrated here. This couple had 4 children as well, and 3 of the 4 of them are affected, and then 1 is not. So now the second mode of inheritance is autosomal recessive, and this is seen in 1 or a few generations, but not multiple, and it can skip generations as a result of that. So again, we'll go through the key here. Again, squares are males, circles are females. But in this case, if an individual has 2 copies of the dominant allele, they're going to be normal. If they have 1 copy of the dominant allele and 1 copy of the recessive allele, they're going to be what's called a carrier. So they don't have the disease, but they have the potential to pass it on to their children. And then if someone is homozygous recessive, they will be affected, meaning they will have the disease. So they have to inherit a recessive allele from both parents. And so if we go to the key here, a carrier is going to be shown by these individuals that have these black stripes like this, so partially filled in. And then if they're fully filled in black, that means they're affected, meaning they're homozygous recessive. Both genders are affected equally in this, and this is often due to consanguinity, which is where you have the mating of related individuals. And this is relevant because let's say you have a family that has a disease, you're going to have a lot of family members that will be carriers. And so if 2 carriers mate, then that dramatically increases the chance of one of the children inheriting the disorder. And so you can actually see that here. So You have 2 carriers here, and then you can see one of their children was affected. Now, again, as we said, in order for the offspring to manifest the disease, it has to inherit 1 copy of the disease allele from each parent. So in this case, you have to inherit the recessive allele from dad and from mom. And again, one of them could be a carrier and one could be affected. Both could be carriers or both could be affected. Doesn't matter. So let's go through some scenarios here that are illustrated in the pedigree. So we'll start here at the top. Let's say one parent is a carrier, as we show here and here, and then the other parent is normal, as we see here and here. And so let's draw out our Punnett square just to illustrate these percentages again. And so as you can see, half of the children will be normal or healthy, and then half will be carriers. And you can see that here. So this couple had 2 children. One's a carrier, and then one is healthy. And then the same thing here. One is a carrier, and one is healthy. So half and half. Now, what happens if both parents are carriers, as we can see here? So both parents are carriers. So let's again draw the Punnett square here. And then as you can see here, 25% will be normal because they'll have 2 copies of the dominant allele. And then 25% will be affected because they'll have 2 copies of the recessive allele. And we say that here. So 25% or 1/4 of the children will be affected. And then as well, the chance of being a carrier is 50%. So you can see that here and here. And so you can see that in the breakdown of the 4 children that this couple had. So one child is normal or healthy. So they had both dominant alleles. And then 2 of the children, or half, are carriers. So they have 1 copy of the dominant, 1 copy of the recessive. And then 1 child was affected. And so they have 2 copies of the recessive. Now, let's say we have 1 parent that's affected. We'll show that here. And then they mate with another parent that is a carrier, as we show here. Let's see what the breakdown is here. And so if you look here, half the children will be carriers, and then half will be affected. And we say that here. Chance of having the disease is 50%, being homozygous recessive, and the chance of being a carrier is 50%. You can see that here in the pedigree, half of the children were affected by the disease, and then half are carriers. Then let's say you have 1 parent that's affected, homozygous recessive, as we show here with this son, and they mate with a parent that is completely normal, so they have 2 copies of the dominant allele, as you can imagine. The chance of the children being a carrier, as you can see with the Punnett square here, is 100%. And then if you look here again, they had 4 children, and all 4 of them were carriers of the disorder. Some examples of diseases that exhibit autosomal recessive inheritance are cystic fibrosis, sickle cell anemia, Niemann-Pick disease, galactosemia, beta thalassemia, and then albinism. So now we're going to go through the X-linked modes of inheritance. And the reason they're called X-linked is because the mutated gene is located on the X chromosome. And so you have X-linked recessive and X-linked dominant. We'll start with X-linked recessive. Here, generations are skipped. And then these disorders, they're seen more commonly in males because males require only 1 copy of the mutation to manifest the disease, while females require 2 copies. So again, it's just like autosomal dominant and recessive for females. So females, they need 2 copies of the recessive allele, so lowercase a. If they have only 1 copy, so let's say they have a copy of the dominant allele, and then they have a copy of the recessive allele, meaning they're heterozygous, they will not manifest the disease, they're just considered carriers. And then obviously if they have 2 copies of the dominant allele, they're completely healthy. Versus males, on the other hand, they only have 1 X chromosome, because they have to have a Y. And so if they have just 1 copy of the recessive allele, then they're going to manifest the disease. Now, there's no male-to-male transmission, and the way that occurs is if we look at example here. So let's say we have a father here who is affected, and he has a copy of the recessive allele on his X chromosome. And then we have a mother that is completely healthy. So she has 2 copies of the X chromosome with the dominant allele. And so let's say they mate and they have a son. So the son has to inherit the Y chromosome. And so he can't inherit this recessive allele X chromosome. He's going to get his X chromosome from his mother, which in this case would be either one of these healthy X chromosomes. So there's a 0% chance of him inheriting this disorder from his father, because he has to have the Y chromosome, and then he's going to get his X chromosome from his mother, which in this case, both of her X chromosomes are healthy. So again, 0% chance. So no male-to-male transmission. And then these disorders are often more severe in males. So now let's go through some examples demonstrated in the pedigree. So if the male parent is normal, so that's shown here at the top here, and the female parent is a carrier. So let's look at the Punnett square for that. The female carrier is going to have one dominant allele and then one recessive allele. And then the father will have one dominant allele and his Y chromosome. And so these Punnett squares for X-linked disorders, you want to think of it as essentially half of the Punnett square is dedicated if the child is a daughter, and then half of it's dedicated to if the child is a son and what the outcome will be. And so for the daughters, if you look here, they have to inherit this X chromosome from their father, so they're automatically going to inherit one dominant allele no matter what. And then from the mother, they can either inherit a dominant allele or a recessive allele. So they have a 50% chance of being completely normal or 50% chance of being a carrier. Now for the sons, they have to inherit the Y chromosome from their father. So they're either going to get this dominant allele from their mother, or they'll get this recessive allele from their mother. So 50% chance of being normal or 50% chance of being affected by the disease. And so we say that here, half the daughters will be carriers, and you can see that with the pedigree here. They had two daughters, one's a carrier, one's healthy, and then half the sons will be affected. And again here, they had two sons, one is healthy and one is affected. So if we look at this scenario here, where the father is affected and then the mother is normal and is not a carrier, let's take a look here at the Punnett square for this. So the mother has two copies of the dominant allele because she's not a carrier and she's completely healthy. And then the father has a copy of the recessive allele along with his Y chromosome. And so let's look at the outcome if the child is a daughter. So they have to inherit this X chromosome from their father. So they're automatically going to get at least one recessive allele. And in this case, their mother is healthy, so they're always going to inherit a dominant allele from their mother. And so as you can see here, all of the daughters will be carriers. Now for the sons, they have to inherit this Y chromosome from their father no matter what. And then they can get a dominant allele from their mother all of the time because their mother has two copies of the dominant allele. So all of the sons will be completely healthy. And you can see that here with the outcome. They had two daughters, both are carriers. They had two sons, both are affected. Now let's go to another scenario here. If the male parent is affected, meaning he has a copy of the recessive allele along with his Y chromosome. And then the female parent is a carrier, so she has one dominant allele and one recessive allele. What will happen is if we look at the daughters, so the daughters, they have to inherit an X chromosome from their father, and so they're going to inherit at least one recessive allele no matter what. And so what happens is they have a 50% chance of inheriting a dominant allele from their mother and a 50% chance of inheriting a recessive allele from their mother. So as a result of that, they have a 50% chance of being a carrier and then 50% chance of being affected. For the sons, they have to inherit the Y chromosome from their father. And then from their mother, same thing. They have a 50% chance of being completely normal because they have a 50% chance of inheriting a dominant allele from their mother. And then they have a 50% chance of inheriting a recessive allele from their mother, which would make a 50% chance of being affected. So if you look at the breakdown here, half of the daughters will be carriers, half will be affected, and then half the sons will be normal, and then half the sons will be affected. Examples of diseases that exhibit X-linked recessive inheritance is Duchenne's muscular dystrophy, glucose 6-phosphate dehydrogenase deficiency, hemophilia, color blindness, and Lesch-9 syndrome. All right, so now if we talk about X-linked dominant, so here we're just going to give you a basic overview of X-linked dominant. We actually have an entire lecture dedicated to X-linked dominant disorders later on in the unit. So for X-linked dominant disorders, it's seen in multiple generations, so it usually does not skip generations. It's more commonly seen in females, as they have two chances to inherit due to having two X chromosomes versus males, which only have one X chromosome. And again, let's look at this here. So for females, all they have to do is get one dominant allele, even if they have a copy of the recessive allele. So let's say we have a father here and then a mother, and they have a daughter. They have to get an X chromosome from their father, and they have to get an X chromosome from their mother. So if either a parent is affected, they have a chance of inheriting that affected X chromosome as a result of that. Versus if they had a son, the sons have to get the Y chromosome from their father. So if their father is affected, they are not going to inherit the disorder. Versus if their mother has it, they have to get an X chromosome from their mother. So it's dependent on if their mother is affected or not. So let's run through some scenarios here in the pedigree. So if the male parent is normal in this couple here, and then the female parent is affected, is heterozygous. So the male is going to have one recessive allele with his Y chromosome, and the female is going to have one dominant and one recessive. And so if we look at the outcome, if the child is a daughter, as you can see here, she's going to inherit at least one normal recessive allele, and then she has a 50% chance of inheriting an affected, which would be the dominant allele, and a 50% chance of inheriting the recessive allele from her mother. So again, 50% chance of being affected and 50% chance of being completely normal. For the sons, they have to inherit the Y chromosome from their father. And so again, they, as well, like the daughters, have a 50% chance of inheriting the dominant allele from their mother, and then a 50% chance of inheriting the recessive allele from their mother. So 50% chance of being affected as well, as we say here. And you can see that they had two daughters, one's affected and one's not. They had two sons, one's affected and one is healthy. So if we look at this scenario here, where you have where the mother is homozygous recessive, so the mother is normal, and then the father is affected, so he has one copy of the dominant allele with his Y chromosome. If we look at the outcome for daughters, no matter what, they have to inherit an X chromosome from their father, so no matter what, they're going to inherit a dominant allele. And so they have this and this. And then from their mother, they're always going to inherit a recessive allele. And so as a result of that, all of the daughters in this case will be affected. Now the sons, they have to inherit a Y chromosome from their father, so there's no way they can inherit this dominant allele from their father. And then their mother has two copies of the recessive allele, so they're always going to inherit the recessive allele, and so all of the sons will be normal. And you can see that here. They had two daughters, both were affected, and then they had two sons here, and both were healthy. Some examples of diseases that exhibit X-linked dominant inheritance are Fragile X syndrome, Alport's disease, Rett syndrome, and hypophosphatemic rickets. So the last of the modes of inheritance that we'll talk about is mitochondrial inheritance, and this refers to the inheritance of mitochondrial DNA, because remember, mitochondria have their own DNA, and this is transmitted through the DNA that's actually found in the mitochondria, which has a circular shape to it. And what's really important is that sperm do not carry mitochondrial DNA. So all mitochondrial DNA is inherited from the mother, and you'll see why in the pedigree that's really important. Mitochondrial inheritance displays variable expressivity and heteroplasmy, and then both males and females are affected by mitochondrial diseases that are inherited this way. And then all offspring from the same mother are affected as a result of that. And that goes back to the fact that sperm do not carry mitochondrial DNA. So if you look in the pedigree here, we have an affected mother mating with a normal father. And as you can see, all four of their children were affected as a result of that. Because regardless if it's a daughter or a son, they have to inherit their mitochondrial DNA from the mother. Same thing here. One of these affected daughters mated with a normal father. And again, all of her children as well also inherited the disorder on the mitochondrial DNA, because again, mitochondrial DNA is only inherited from the mother. On the flip side here, you had an affected son who mated with a normal female. And since she is normal, this is where all of her children are going to inherit their mitochondrial DNA. So in this case, all of the children inherited normal mitochondrial DNA. Very important to remember for mitochondrial inheritance. So these mitochondrial diseases, they result in failure of energy metabolism due to dysfunction of the mitochondria. Remember, the mitochondria is essentially the powerhouse of the cell. And so this really affects tissues that have high energy requirements, such as muscles and the central nervous system. Some examples are Leber's optic neuropathy, myoclonic epilepsy with ragged red fibers, and then MELOS, which refers to mitochondrial encephalopathy lactic acidosis, and stroke-like episodes. All right, and to close out this lecture, we're going to talk about Hardy-Weinberg equilibrium. So there's 4 assumptions you have to make for the Hardy-Weinberg equilibrium. 1, that there's random mating occurring, there's no migration, no natural selection, and then no mutations occurring. And so these are the 2 equations that you're going to use. This one, the first one here is for genotype frequency, p squared plus 2pq plus q squared equals 1, and then allele frequency for p plus q equals 1. So what do each of these mean? So p equals the frequency of the first, usually normal allele, often assumed to be 1 in the normal population. So that would be this guy right here. Q is the frequency of the second allele, usually the disease. P squared is the frequency of a genotype homozygous for normal. So let's say this was autosomal recessive. Just use that as an example to illustrate this. So this would be the frequency of homozygous dominant. Meaning AA. And then Q squared over here is the frequency of genotype 2, which is homozygous for the disease, or the disease prevalence. And so this would be homozygous recessive, AA. And then 2PQ is the frequency of genotype 1 and 2 in combination, also known as heterozygous, or the carrier in the case of autosomal recessive for the disease. So this would be AA. It's important to note that in males, the frequency of an X-linked recessive disease is simply equal to Q, not Q squared, because males only have 1 X chromosome. And then as a result of that, they only need 1 copy of the recessive allele to exhibit the disorder. In females, the frequency of an X-linked recessive disease is similar to other types of diseases, and so you would just have it equal to Q squared. So the way you'll get tested on Hardy-Weinberg equilibrium is not really to regurgitate any of the definitions we just went over, the equations. It's going to be really applying those equations and concepts to an example problem. And so this is an example problem that's in your book. I'll give you a second here if you want to go through it, and then we'll walk through the answer. So they're asking, if the prevalence of an autosomal recessive disease is 1 in a million people, what is the frequency of carriers? So let's look at this here. What do they tell us? So the disease prevalence is 1 in a million, which is going to be equal to Q squared. So remember, we have 2p squared plus 2pq, and this is the genotype frequency, is equal to 1. They tell us it's an autosomal recessive, so this is going to be homozygous dominant. This is going to be the heterozygous. This will be homozygous recessive. So they're telling us that the homozygous recessive, or the population of the disease, the prevalence of that is 1 in a million. So they're telling us that Q squared is equal to 1 in a million. So if we take the square root of this to give us Q, that's 1 in 1,000. Now, the question is asking for the frequency of carriers, which would be those that are heterozygous. So that would be what 2pq is equal to. So we have to use this to figure out 1. So remember, p plus q equals 1 for the allelic frequency. Now, in this case, 1 over 1,000 is obviously very small, and if you subtract that from 1, it's going to be about 1. And the other thing here is that, as we said, p can be assumed to be 1 for very rare diseases, such as diseases that occur about 1 in a million people. And so you can assume p is 1, so 2pq is going to be equal to 2 times 1 times 1 in 1,000. And so that's going to result in the answer of 2 over 1,000, or 1 in 500. So the prevalence of carriers in the population is 1 in 500. All right, so that closes out our discussion of the different modes of inheritance and the Hardy-Weinberg equilibrium. And now in the subsequent lectures, we're going to apply these genetic principles to genetic disorders you may see on your exams.